Hey, all right. So we've done male lab, we've done female lab, so now we've got to do pregnancy lab. I don't go into just a whole lot of detail about this. I'm just going to kind of give you some highlights on the structures that you need to recognize um, for the exam. There's also the video by Miss Graham. She does a little more detail, but again, I kind of keep it light. There's not going to be a whole lot of questions from the pregnancy models on your exam, so don't stress too much. So what we have in lab are some pregnancy models, and we start from basically fertilization all the way through um, full development and delivery. And so when you look at our pregnancy models, I'm going to show you some different stages that we have. We actually have more than what I'm showing you in the video because, again, I just don't get into that much detail with my students. So if you're watching Miss Graham's and she goes into more detail, that's okay. The more you know, knowledge is good. But for me and for my exam, it won't be that much detail. So I'm going to basically cover these different structures for you. So before I do, what I'm going to show you are going to be some pictures from your textbook and then try to relate that to the models so that you can kind of have a picture of, oh, this is that, this is that. So just like I do with most of your lab videos. So when you have the sperm, okay, here's the sperm. So we're in the uterus. Here's the fallopian tubes. The sperm have been swimming up and they're going to meet the egg right here, okay? So we have a fertilized egg, which we call the zygote, remember? All right, that's is going to divide and divide and divide. So it'll divide into 2 and then to 4, and then 4 becomes 8, and 8 becomes 16, and 16 becomes 32. 32 becomes 64. I think in another video, I said it became 70 something. Anyway, math is hard, but you get the idea. So double, 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 double. So finally it becomes just a mass of cells called a morula. And this morula will then become what's called the early blastocyst. And the blastocyst is where you're going to start having a fluid-filled cavity inside of this group of cells. Now, what's really important between the zygote stage and the morula stage is all of these cells are dividing and dividing and dividing, but the overall structure is not getting any bigger. Okay, because we don't want it to get stuck in the fallopian tube. Make sense? Yeah. So it doesn't really start to get bigger until after it implants. So you're going to have this early blastocyst. That's going to implant into the uterus. Then you're going to start to have more mitosis in the cell structures. The overall structure will get bigger and bigger and bigger. But until it actually implants, it pretty much stays the same size. You just get more and more cells dividing inside that structure. So again, it doesn't get stuck in the fallopian tube. Just kind of makes sense. So what are you seeing in the model? Well, here we have that ovulated egg. So here you have your egg, and remember you have the corona radiata that's around the ovum, and the sperm has to break its way through there. So here you have, let me see if I can change colors a little bit. Let's go to red. Here you have the sperm breaking through the corona radiata. So here would be your oocyte with your corona radiata and your sperm, right? Well, here we have the sperm, which is broken through the corona radiata. Remember, it goes through that cortical chain, so it kind of hardens up so no more sperm can get in. This is kind of the stage we're at now. And now you have the DNA from the mom and the father going together, and you're getting fertilization. You'll also notice that this is going to cause the ovum to continue with meiosis, and you're going to get those additional polar bodies. All right. Now, once fertilization has occurred, you have this fertilized egg, which now is going to be a zygote. So this is going to be just your one-celled zygote stage. You're going to get division, 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 division. So this is going to be the four-cell stage. It may actually be the 8-cell stage, but you really can't see the other 4 because of the way it's cut. So we're just going to call it the 4-cell stage. My model's my rules because you can't really tell. Then it's going to divide, 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 divide. And on this one, this is actually going to be, there's actually 16 cells in this one. You can really see where it's divided many, many times. And we don't really have a morula. So usually what I say on this one is I just call this the morula stage because it's just a really big mass of cells because it's hard for you guys to actually see that it's 16. So if you see a on this particular model, it looks like a bunch of cells filling up that little capsule. We're just going to call that the morula stage. Again, we've got several models. So I'm just trying to simplify it for you. So this is going to be the 1-cell zygote, the 4-cell, and this will be the morula stage. Okay? Make it easy. All right, so we've got that morula stage. So the next thing we want to do is it's going to have to hollow out and form a blastocyst so that it can implant. So here at a week, we've got that implantation occurring. All right, so remember back to when we talked about what happens with implantation and placentation. You start to get the formation of the syncytial trophoblast, or where you're basically sending out those cells into the placenta or into the endometrium that are going to form the placenta. You're seeing the first divisions, first differentiations of those cells, where you're getting an early yolk sac. You're getting the beginnings of the baby here. You're getting the amnion here. All right. We have full implantation here. You can see we're beginning to get the yolk sac here. Again, early divisions of the blastic tissue, the endoderm, ectoderm, and mesoderm are starting to form. Not quite there yet, but they're getting there. More fully formed tissues going out into the endometrium to form the placenta. And at 16 days, this is really where we can really start to see some differentiation. We can see a fully formed yolk sac. We can see an endoderm, a mesoderm, and an ectoderm. We can see an amniotic cavity. We can see the amnion. We can see the chorion, which is this kind of circular material here. And we're starting to see the beginnings of the maternal blood vessels start to infiltrate and surround the chorion. So I'm just going to have 2 models to show you on this one. So here's going to be that early blastocyst, and you can see the first cavity here, the beginnings of what's going to become the baby in this particular area. So this is going to be the embryo. And then you have the beginnings of the chorion. Okay, so it's just starting to infiltrate out into the endometrium, and it's going to become the chorion. It's kind of this early chorion. But in this I'm so sorry, I'm so sick. In this slide, you have your 3 embryonic membranes are clearly visible. You have your yolk sac, you have your amnion, and then you clearly have your chorion. And that's really all I would ask you on this particular slide, is to identify those three membranes. Now, once you get up to about 4.5 weeks, you can see even more stuff. You can still see your amnion, you can see your chorion, you can see your yolk sac, and you can see the chorionic villus and the decidua basalis, which is going to kind of form the mother's end of the placenta, with the maternal vessels kind of wrapping around those chorionic villi. So on this model, here's your amniotic membrane. So this is your amniotic cavity. Here's a developing embryo. You can see a little heart there. Here's the yolk sac. Here's the chorionic villi, kind of invaginating up into this maternal blood sinuses here. And you can see the beginnings of what's going to become the umbilical cord. All right. This particular model, you can see the yolk sac, you can see the amniotic cavity, this white membrane's the amnion, and the beginnings of the formation of the embryo, and you can see the chorionic villi. Then you get to the mature uterus and the mature fetus. You're kind of at the 13-week stage where you're no longer an embryo. All your organ systems are there. And so you can see your amnion again. You can see a little bit of the yolk sac left, fully formed umbilical cord, and a fully formed placenta. So what that looks like, here's your placenta. Here is your amnion wrapping wrapping around this cavity so the baby's floating in that sac. You have your uterus here and beginnings of an umbilical cord. Blow that up to the very end, so this is almost time for delivery. You can see your fetus laying in there. There's an umbilical cord that's kind of hanging off. You can't really see it the way the baby's turned. You have your placenta. You have your cervix. And literally, that's all I'm going to ask you. I just don't ask a whole lot of questions about the pregnant models. Just very simplistic. Be able to recognize the membranes, the chorion, the amnion, the yolk sac. Be able to recognize the umbilical cord, the placenta, and really that's about it. Okay, so watch Miss Graham's video. Again, she goes into way more detail than I do, but that should at least give you an idea of where to start, and expect a couple of questions from the pregnant models on your exam. Thanks.